DNA is a large molecule made up of a long sequence of 4 nucleotides: adenine, A, cytosine, C, thymine, T, and guanine, G. In humans, we have 46 of these DNA molecules, organized into 46 chromosomes, and each one contains thousands of genes that carry instructions for traits like eye color, hair type, and even the risk of certain genetic disorders. Sometimes, a genetic disorder can result from a mutation in just one gene, and to detect this specific change in the long DNA sequence, we use a technique called gel electrophoresis. In gel electrophoresis, the first step is to cut the DNA into smaller pieces using restriction enzymes. These are special proteins that slice DNA at certain nucleotide patterns. After cutting, the DNA pieces are placed into small wells in a slab of agarose gel. The gel might look solid, but it's actually semi-solid. And under a microscope, it looks like a maze filled with water. A negative charge is applied near the wells, and a positive charge is placed at the opposite end of the gel. When an electric current flows through the gel, it pulls the DNA pieces, which carry a negative charge, toward the positive side, guiding them through the gel's maze-like structure. What's important is that smaller DNA fragments move faster and travel farther through the gel than larger ones. Now, in a perfect world, if we could easily recognize a mutation, things would be simple. Sometimes spotting a mutation isn't that simple, but this is where restriction enzymes become really useful. For example, if we use a restriction enzyme called EcoRI to cut the DNA, it will attach to every GAATTC sequence and cut between the G and the first A. So, if we have a DNA strand with 3G, AATTC sites, EcoRI will cut at all 3 points, creating 4 DNA fragments. When we place this DNA into a well in an agarose gel and run an electric current through it, we'll see 4 bands on the gel. Each band stands for a fragment of different size, showing us that 3 cut sites were present in the DNA. What's important to note is that restriction enzymes can cut any DNA as long as the specific sequence they recognize is there. However, how DNA is cut can differ from person to person because everyone's DNA sequence is slightly different. That's why the sizes of the DNA fragments can also vary among individuals. This kind of variation is called polymorphism, so we call these fragment differences restriction fragment length polymorphisms. If the variation happens at a spot where a restriction enzyme cuts, we call that a polymorphic marker, because it marks a known location on the DNA where differences can be detected using that enzyme. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. You see, since restriction enzymes cut DNA at specific nucleotide sequences, they won't cut the DNA if there's a mutation in that exact sequence. For instance, imagine there's a change in just one nucleotide. This is called a single nucleotide polymorphism. Now, suppose that this mutation happens right in the binding site of a restriction enzyme, like here, where a single A is replaced by a G. That change would affect the middle binding site for EcoRI, stopping the enzyme from attaching and cutting at that spot. As a result, after using eco.RI, the DNA breaks into different fragments. Now, instead of 4 fragments, we end up with only 3, and the middle piece is longer than before. On a gel electrophoresis, we'll see just 3 bands, showing that a mutation is present in the DNA. Another kind of polymorphism is called a short tandem repeat polymorphism, also known as a microsatellite. An STRP happens when a specific DNA pattern, like a CAT trinucleotide, is repeated man times in a row, often between 5 to 50 times. These STRPs matter because their repeated nature can cause problems during DNA replication. To put it simply, the enzyme that copies DNA, called DNA polymerase, can sometimes slip and copy the CAT repeat more times or fewer times than it should. This changes the length of the allele, making it longer or shorter than the original. And again, if we cut the DNA with a restriction enzyme, then the piece with more repeats will be longer than the piece with fewer repeats. These differences in fragment sizes can easily be seen using gel electrophoresis. So, gel electrophoresis helps us see DNA mutations and DNA repeats, and the main idea is to use these features as markers to track genes we care about. Remember, our 46 chromosomes are arranged in 23 pairs of homologous chromosomes. When gametes, like sperm and egg, are made, these paired chromosomes swap genetic material through a process called crossing over. In crossing over, the free ends of 2 homologous chromosomes twist and exchange segments. This is great because it increases genetic diversity, but it also makes it harder to trace specific genes, as they can move between chromosomes. Luckily, crossing over follows the rule of genetic linkage, which says that genes located close together on a chromosome are less likely to be split apart during crossing over. So, if our marker and a mutant gene are very close on the chromosome, they will likely stay together during recombination. For instance, if there's a known SNP single nucleotide polymorphism near or inside a gene, and we can detect this SNP using gel electrophoresis, then it's a strong hint that the gene of interest is also present in that individual. To really make the most of gel electrophoresis, the key is figuring out how close a marker is to the gene of interest. One way to do this is by studying a family where some members have the disease, and then creating a pedigree chart. Next, we test each family member for known markers, and then look at how those markers relate to who has the disease. Let's say we have 4 markers, and for simplicity, all 4 are SNPs, small point mutations at different positions in the DNA. Looking at the pedigree, we see that marker 2 matches up best with the disease. It appears in all 7 individuals who have the condition, but it also shows up in 2 healthy people. This tells us that those 2 people inherited the SNP, but not the disease-causing gene. So, in 2 out of 9 cases, crossing over separated the marker from the mutant gene, meaning marker 2 stayed linked to the mutation in 7 out of 9 cases. That makes marker 2 the best choice here. It's likely the closest to the gene causing the disease. In real genetic studies, pedigrees are tested with hundreds of markers across the whole genome. Then, researchers pick the closest one, which becomes the most useful marker for tracking the gene. All right, here's a quick summary. Genetic testing uses different techniques to help locate and identify the mutant allele responsible for a disease. First, we need to track down the mutant allele by using genetic markers that stay linked to it during crossing over in meiosis. These markers can be SNPs or STRPs. Then a pedigree chart is created for the affected family, and family members are tested to find the most reliable marker. How well a marker stays linked with the disease gene tells us how close it is to the mutant allele. The closer the marker, the better it works for tracking the gene.